to welcome you all to the CIBM Center for Biomedical Imaging Breakfast and Science Seminar Series. We're at number 13 now, and I'm very happy to welcome newcomers today from Amsterdam and Leeds University Medical Center as well. So without further ado, I would like to invite Matthias Stuber, who is the head of the CIBM MRI CHUV Unit Translational MR Imaging Section, to introduce the speaker today, who is Ruud van Heeswijk. So Matthias, the floor is yours. I'll stop sharing my screen.
Thank you so much for joining this exciting event. It's my pleasure to introduce Ruud van Heeswijk, and here I have his parcours, as it were, in a nutshell. He did his master at the Eindhoven University of Technology in 2004, and during his studies he actually did a short stint at Johns Hopkins, where strangely enough I've never met him because we were there at the same time. Then he did his PhD at the EPFL in 2009 with Rolf Grüder. Then he joined our team as a postdoc at CIBM MRI CHUV UNIL from 2010 to 2014. Here you can see the first team, or a younger rendition of that team anyway. Then he became a lecturer or maître assistant in 2014, again at CIBM MRI CHUV UNIL. Then he became a privat docent in 2017, before becoming a senior lecturer or MER in 2018. So Ruud is actually quite decorated. He won the Pfizer Basic Cardiology Research Award in 2014, which is very, very prestigious. So we went to Zurich to accept that award, or at least I was gloating he was accepting the award. Then he won the Hossein Sadeghi Prize for Outstanding Cardiovascular Research in 2015. And he has been awarded many, many grants. And I don't know whether he knows it, but I have an Excel spreadsheet. So to the tune of more than 1.5 million Swiss francs, he has been getting grants, among which an Ambizione grant, which really jump-started his career with his own PhD student. And then he was awarded the project grant just 2 years ago. He already has had 2 PhD students where he was the thesis director, who successfully defended their PhD thesis. He is now on the program committee of the ISMRM and is the educational chair for the cardiovascular section in 2022. He is an SCMR fellow and on the editorial board of JCMR. And I think he has about 50 peer-reviewed publications under his belt. And I think it's probably fair to say that Ruud is one of the CIBM success stories, moving from one CIBM section to enrich the other, and graduating from PhD student to becoming an accomplished and decorated principal investigator. And with that, I would like to, here you see his first 2 PhD students actually sitting at the scanner together with him. And with that, I would like to hand over to Ruud van Heeswijk, and I'm looking very much forward to his presentation, where he will enlighten us on the use of toothpaste, I believe.
Thanks for the kind introduction to Matthias. Made me blush a little bit. Thanks a lot, Pina and the CIBM, for the invitation to present my work over here. So I'm going to be talking about inflammation imaging with fluorine-19 MRI. So not from bench to bedside, but from toothpaste, to be very specific this time, because probably when people think about fluorine, if they don't know this type of MRI, they may think of toothpaste and teeth and so on. Luckily, we're not going to talk about toothpaste too much, but I had to put it in, of course. So as Matthias already said, I've done quite a lot of different things in MR over the past. And these days, though, I'm kind of more limited to 2 research themes. One is indeed fluorine-19 MRI, which is mostly preclinical, is a type of molecular imaging, makes use of nanoparticles. And I mostly use it for inflammation mapping. However, I also do a lot of relaxation time mapping, so T1 and T2 mapping and so on, calculating physiological values in each pixel of an image. And yeah, this is mostly clinical these days. The novelties mostly come from physics and mathematics. And I mostly use it to detect fibrosis and edema. But so I will not be talking about this second theme today, but just in case you're looking to develop or apply something of it yourself, well, after this session, you should know my email address, so please contact me if you want to. So Fluorine-19 MRI today. So what's on the menu for today? First, we're going to look at what it is, Fluorine MRI, and how we do it. Then we will take a look at several preclinical applications, so mouse studies, rat studies, and this kind of thing, and to see what you can specifically do with it. Then we'll take a look at some translations. So if you want to move towards clinical application, which I guess in many MR developments and imaging developments is the final goal. We want to see what hurdles we encounter and how we can overcome them. And finally, we want to look at some early examples of clinical applications. So I'm going to remove my, try to move my bar of Zoom a little bit. So first, let's look a little bit at the background of Fluorine-19. Actually, there is no background to Fluorine-19, so let's see why. So first of all, toothpaste, as I said. So when you think of toothpaste and fluorine, you actually think of fluoride, which is a sodium fluoride. And indeed, there's 0.35% or so of fluoride in toothpaste, and that's because it strengthens your enamel. And if you indeed put a tube of toothpaste like this in a Fluorine RF coil, like I did when I just received this coil just to see if it works, you can actually see that, yep, you get a peak and you get a signal. If you do an unlocalized FID, so you just send in an RF pulse and see if there's some signal coming back, you get a peak, so confirming that this RF coil works and that there is indeed fluorine in toothpaste. So that's all I'm going to talk about toothpaste-wise, though. So what is fluorine? It's over here in the corner in the periodic table, and it's actually been used from the very beginning of NMR and MRI. This is an imaging experiment that Holland and Bottomley published in 1977, before several of us were born even. And indeed, it's never really gone away. However, it made a revival in the last 10, 15 years, though, and I'm going to tell you why it made this revival. One of the main reasons is that it's 100% naturally abundant. So, of course, we all know fluorine-18, which is radioactive and is used for positron emission tomography, for PET imaging. But if you look in nature, all fluorine is basically fluorine-19. So that's handy for us. And it is a very high sensitivity compared to other X nuclei. So compared to, say, carbon or phosphor and so on, it has 85% of the sensitivity and 95% of the resonance frequency. So it's fairly easy, say. It has also quite a long relaxation time, so there's no funny decouplings or whatever you need to do to get rid of very short relaxation times. So it's fairly straightforward to do Fluorine-19 MRI. And it has, very luckily, no occurrence in the body. So if you make a normal MR image of a human body at the fluorine-19 frequency, you won't see anything. That's because the little bit of fluorine that's in the body is actually sequestered, not moving in the bones and teeth. So that's not MR detectable, at least with most normal sequences. Perhaps these days with UTE and this kind of thing it is. But for most regular MRI, we don't see it. That means we need to inject something. And that's cool, because then it means that any fluorine you inject can kind of be handled as a tracer. So anything you see is the thing you injected. So what that means is that it goes a little bit like PET MR. So this is an image that Matthias made for a long time ago, which resembles this one from even longer ago. And then I added my art to this. So this is what a fluorine part would look on after you injected it. And then you can, of course, overlay the two, and then you can see where, for example, your inflammation would come from, if this would go to inflammation or whatever other reason you have to follow it. But so this is very much like PET-CT, as you've probably seen before. PET, positron emission tomography, is much more sensitive than MR. They can detect on the pico scale, picomolar, while we're at the micro to millimolar scale. However, fluorine MRI has several unique advantages of its own, which I'll go over in a bit. However, what do we inject with fluorine-19 MRI? And you might remember this movie from, I think, the late 80s, early 90s from, what's his name, James Cameron, who also did Terminator and Avatar and this kind of stuff. And here, some people went deep sea diving, but really like kilometers under the sea. And to go diving in that pressure, the main character, Ed Harris here, he had to breathe some kind of liquid because the pressure was too high in the movie, etc., etc. And that sounded like science fiction, of course, to breathe these liquids, but actually that's reality. So here's a mouse inside the perfluorocarbon, and it's happily breathing away. And that's because perfluorocarbons, when they're liquid, they have a 4 times higher concentration of air than actual air, of oxygen than actual air does. So perfluorocarbons, they can take up a huge amount of oxygen, and they've actually been already injected in humans, up to ½ liter of them, as a blood volume replacement. It's even more funny because then, of course, you think oxygen, blood, and so on. What do you think about immediately? Probably cycling. So, of course, cyclists immediately jumped on the occasion when it was discovered as well. And that was one of the first times, I guess, that the CHUV made it to the New York Times, because a cyclist used perfluorocarbons as doping to increase the oxygenation of their blood, and then they used a little bit too much. And that was already back in the late 90s. So its use has been quite well known, but we're not going to focus much on these uses. We're going to look at it from the inflammation point of view. So why can we inject so much perfluorocarbon? Well, mostly because the fluorine carbon bond is the strongest single organic bond. So it's chemically inert, it's very stable and completely non-toxic. And I said before, it's already been in phase 3 and now I think even phase 4 trials as a blood volume expander. So it's been used up to ½ liter or so in with up to ½ liter in humans. And that was especially in the 80s or so as this blood volume expander. These days is mostly used for other things like oxygenation of critical areas such as brain trauma. But yeah, the main point I want to get across here is that it's indeed very, very safe, even in large volumes. And several examples, as you can see here, look like regular hydrocarbons. Just all hydrogens have been replaced with fluorine. So the most common ones are perfluorooctylbromide. The main thing here that there's a bromide atom attached here, which makes all these fluorines slightly magnetically inequivalent. There's this one, the crown ether, and there's a perfluoropolyether. So this is a polymer that with a short chain length here, which has mostly a single resonance that we're interested in, but we'll look at them a little bit later. And what we do with them, PFCs are both lipophobic and hydrophobic. So we put them in emulsions that circulate a little bit longer. If you inject them pure, there's no harm done, but you almost immediately breathe them out. So you mostly eliminate perfluorocarbons through the lungs. And if you want to keep them around a little bit longer, and if you want to have them taken up by the immune system, you need to put them in some particles. So what you do is you put some water and perfluorocarbon together, then you add some surfactant like lecithin, comes from chicken eggs usually, and then you do a high pressure homogenization and you get like a milky white liquid like this that's heavier than water. And that mostly consists then, if you put it through a filter, of particles of your filter size. So these phospholipids from the surfactant then enclose the perfluorocarbon inside and the water will be outside. So what happens then with these surfactants? Well, you inject them intravenously, in humans usually in the arm, in animals, such as mice and rats, usually in the tail vein. And once you inject them in the vessel, the particles go around the blood, in the bloodstream. They circulate until they're breathed out, etc. If they are near immune cells, then they are exactly the right size to pique the interest of immune cells, so they get phagocytosed. And then if you wait a short while, usually about an hour or two, they're cleared from the blood circulation itself. They'll still be in all other repositories of immune cells, such as the liver and the spleen, etc., but they'll be gone from the blood. And these immune cells will have traveled to the site of inflammation, as you see here. So the nice thing is then, of course, that after they're cleared from the blood, the signal now only comes from the site of inflammation or site repositories of immune cells. And that means that we have a nice high specificity. We don't need to compare to neighboring tissues or any changes in contrast like with regular MR. It's directly, if there's a signal somewhere, then it will be made by the perfluorocarbons injected. So if there's a brighter signal, that means that there are more perfluorocarbons there. If there's a larger volume, the inflammation is probably wider spread. One unique property of fluorine compared to, say, PET or iron oxide is that the perfluorocarbon concentration reflects immune cell presence over time. So if the immune cell goes away or dies, the perfluorocarbon is released from the site and the signal also disappears. And that's quite unique. And that was very elegantly demonstrated by the Düsseldorf group of Uli Vleugel a few years ago. So here they injected an irritant in the fat pad on the back of some rats. As you can see over here, here's that fat pad, and it's full of, or it's injected with irritant here. Here you see the wound itself, it still looks a bit inflamed. And indeed, the Fluorine MRI shows a large patch of inflammation. And then they monitored this over time as the inflammation was cleared out, the wound indeed closed, and the redness went away. The fluorine signal also slowly decreased. And if you track the three of them over time, so here's the irritant decreasing over time, here's the fluorine signal over time, and the immune cell concentration that they took out by assay, you can see that it's exactly the same curve shape. So you can very nicely use fluorine MRI to monitor inflammation over time. Other uses that there are for fluorine MRI. So you, of course, what we do here is indeed injecting a perfluorocarbon emulsion into the tail, and then it's taken up by macrophages and other immune cells in the body, and then we can scan it and have a perfluorocarbon hotspot somewhere in the animal. Or we could take out an individual organ, then liberate all the cells, and then do MR spectroscopy, and so get an inflammation index basically of different organs. That's another option. But what we can also do is take some ex vivo or in vitro cells and label those in a culture with perfluorocarbon, and after washing those cells so that there's only perfluorocarbon on the inside, we can inject those labeled cells into the mouse or rat, and then track those over time. So we also do hotspot imaging, but now we will have specifically tracked the cell. I don't do too much of this myself, but it's definitely a fairly straightforward possibility. And of course, the labeled organs or the organs that are labeled by these pre-labeled cells can of course be isolated and quantified in the same manner as I mentioned before. And the final option, but the third option is indeed that we have a multimodal particle. So instead of just injecting it and hoping the immune cells take it up, we can attach a ligand that targets, for example, neovascularization, like alpha V beta 3 over here, and have the perfluorocarbon particle targeted to a specific disease or biological process. At the same time, since we have this layer on the outside, and this lipid layer, we can also attach fluorophores so that we can do, for example, immunofluorescent histology afterwards, or similar other processes with other markers. So overall, the main advantages of fluorine MRI is that there is no background signal. It's highly specific. Because of this, the signal roughly corresponds to the fluorine quantity, so it allows for easy quantification. And the molecules we inject are inert and safe, which should allow for easy translation. And finally, there is a clearance from the tissue, so the molecules don't disappear very quickly by themselves, and neither stay resident forever, so you can use them to monitor things over time. However, there are also clear disadvantages. One, the fluorine nucleus requires special hardware, so a clinical scanner will need a broadband amplifier and specific radiofrequency coils. And the technique, the main challenge is the low signal to noise ratio, so our detection limits are relatively low, meaning that the concentrations that we need to detect the signal are quite high compared to some other molecular imaging options. Now, first, I want to look at some preclinical applications. So some mouse models that I've worked on over the years. And the first one is myocarditis. So this idea came to us when I just started as a postdoc from Jürg Schwitter, who knew another cardiologist, Ulrich Eriksson, in Zurich, and Dr. Eriksson had a very well characterized mouse model of autoimmune myocarditis. So for those of you who don't occupy themselves much with the heart, myocarditis is the inflammation of the heart. It's a kind of a group name, a big collection of things, and it essentially consists of inflammation of the heart muscle. So here you see a diagram of a healthy muscle, and basically these get hindered by the infiltration of many immune cells. You see in the bottom little graph, and it has many causes. You can have viral myocarditis, which is by far the most common. So this is usually in the flu season, when the flu virus gets lost and ends up in the heart, where there are relatively few native immune cells. But it can also happen through toxins, drugs, even lightning strike can cause myocarditis. And the main danger is that if it's not detected early and treated with something anti-inflammatory, that it can lead to permanent dysfunction or what do you call it, deformation of the heart, such as dilated cardiomyopathy, and it can lead to cardiac arrest indeed if untreated. So it's the main cause of cardiac death in young sporting people, for example. So it mainly needs to be diagnosed early. And that's often done with cardiac MR and with echo while there's already dysfunction, or when there is already scar in the heart, for example. And of course, fluorine MRI could be very useful by detecting the inflammation directly before any damage or dysfunction has occurred. So we went ahead and did this. So this is an image of the heart in the short axis. So here you see the left ventricle that does the main pumping to most of the body, and the right ventricle, which is a very, very thin wall in the mouse. But you can still see a fluorine signal on several spots in the wall of this heart. And when we did this ex vivo, we saw indeed that we could confirm these patches of fluorine in the very same mouse. And then when we did histology of the right ventricle, so this wall is really a small part of a millimeter thick, we could indeed see a bit of infiltration of immune cells here in the corner. So the purple are basically cellular nuclei here. So we had histological confirmation in the exact same area. And then very interestingly, when we looked at these mice and looked at their disease score, so how severe the myocarditis was in these animals. So this is a compound score of mostly behavioral and echo parameters. Then we saw that basically the ejection fraction hadn't changed yet. So the functioning of the heart hadn't changed yet as a function of this disease score. So the lower disease score, the function was still the same. However, our signal volume, so how much fluorine there was, and which basically is an indication of the amount of inflammation, that very significantly correlated with the disease score. So we were able to detect inflammation in a myocarditis model in the mouse at a very early stage, and also nicely correlated with the disease score. And this research led to the Pfizer Prize in cardiology that Matthias already mentioned earlier. Then another cardiovascular disease that's very common, or even more common than myocarditis, is inflammation imaging of is atherosclerosis. So we wanted to image the inflammation in here. And atherosclerosis causes most strokes and infarction, and thus it's a major cause of death and has a very high socioeconomic impact. So you're all familiar probably with atherosclerosis, but if not, it's a chronic disease, so slow build-up. Probably some of us are already building up atherosclerotic plaque in several arteries in our body, and it consists of several stages. So first there is some endothelial dysfunction, infiltration of nitric oxide. Then relevant to us, there is a time where immune cells start to infiltrate into the wall. There is no lumen narrowing yet, so the vessel volume doesn't constrict yet. There's no stenosis yet. And after a lot of accumulation of immune cell, fat also starts to accumulate, and the cap, so the layer that separates this infiltrated fat and immune cell mass, at some point it can burst, and then all kinds of trash basically is released into the bloodstream. And these things, these emboli, they can basically be very deadly if they occur in the brain or in the heart. So as with myocarditis, we want to detect atherosclerosis as early as possible. And indeed, we focus here on the inflammation step, because it is now quite well agreed on that the plaque vulnerability, so the degree to which it is prone to rupture, to cause a lot of problems, is regulated by inflammation. So the inflammation most likely drives when plaques will burst. So we wanted to see if in a mouse model we could detect atherosclerosis at an early time. And for this we worked together with the group of Uli Vleugel in Düsseldorf, who developed the perfluorocarbons we used, with the group of Lucia Mazzolai here at the CHUV, who had a very well characterized model of mouse atherosclerosis, and of course with Matthias, who was well, paying me and funding this research. So luckily we could detect the inflammation. So again, this is a mouse aorta, right? This is a few hairs thick, basically, and we could still detect plaque in the carotid. We could see some confounding signal from the isoflurane what do you call it, anesthetic. However, this is mostly limited to some fatty tissues and so on, and to the skin. And then, as with most perfluorocarbon compounds, it mainly accumulates in the liver, so we get a very nice and bright signal in the liver. But also, as you can see here in another mouse, there is clear signal in the aortic arch of the mouse. And then when we did this ex vivo, so we took out these aortas and put them in the 9.4 Tesla at TPFL for an entire night at a resolution of 0.1 millimeter or so. We could even nicely confirm the individual plaques that we see in the aorta in these mice. And the same with some high resolution imaging. When you render it, they could nicely see that indeed in the usual spots where you expect the inflammation, you could clearly see patches of fluorine as well. Then we put in the same particle that we injected, we also put some rhodamine so that we could see the immunofluorescent histology. And then here you see a nice cross section of the plaque. So here you have the artery itself, and then you can see here sitting inside that artery the atherosclerotic plaque. And then inside that plaque you can see a lot of macrophages. And then if you zoom in a little bit further, so the dashed bar here, you can see here that the immunofluorescent histology confirms the presence of the perfluorocarbon inside the immune cells. So just to guide the colors around a little bit, The blue is the cell nuclei, so it just shows that there are cells there. Green is perfluorocarbon, and red is macrophages. So if red and green overlap, basically they become yellow. So all the yellow orangey bits, those are immune cells that also have perfluorocarbons on board. However, there are also perfluorocarbons in some cells that are not macrophages. There might be other immune cells, or there might even be some endothelial cells that have some capability to absorb the nanoparticles. But the main thing to see here indeed is that the macrophages, they do absorb indeed the perfluorocarbon. Then a much more recent project, and that brain people are a bit more familiar with probably, monitoring inflammation in glioblastoma. So this is a collaboration with the group of Johanna Joyce, and mostly done by her PhD student Davide Croci together with me. As you might know, glioblastoma is the most aggressive cancer in the brain, and there is no current treatment that it has any long-term effect. So surgery, chemotherapy, radiation, etc., all lead to a 5-year survival somewhere on the order of 5% or something like this. It has a relatively high occurrence for brain tumors, so it's 15% of all brain tumors or so. And there's a lot of research at the moment to look into the biology of the immune response to these glioblastoma, because new immunotherapies may be developed that can let the body itself attack these tumors. So we first looked at some mice with glioblastoma at our 3 Tesla clinical scanner, so a relatively low sensitivity, but still we've been doing this for a while now, so the clinical scanner should be able to show the same level of inflammation. So we injected some perfluorocarbon, the crown ether in this case, which has very good MR properties, into the tail of some mice with glioblastoma. And there indeed we see what we hoped that we would see. So if you zoom in a little bit, you see in the brain, you see that the tumor indeed highlights. You see the reference tube that we placed on the outside of the mouse, and you see that the liver indeed also generates a signal, because the Kupffer cells indeed, as always, absorb a lot of fluorocarbon as well. Interestingly, there is no one-on-one overlap of the fluorocarbon and the tumor. So this is another mouse that you see here on the right, rendered in 3D. But essentially, with the transparent gray bit is the brain itself. The blue is the quite large glioblastoma, and in this mouse, for example, there was inflammation in only a small part of the tumor, meaning that either there was a much higher activity of the immune cells over there and there was no inflammation active, or the inflammation was a much lower grade in the rest of the tumor, for example. Then there was some biological characterization of the cells in the brain. So again, here we added some rhodamine to the nanoparticles, and there we could indeed see that the perfluorocarbon is again inside the immune cells. So here inside an immortal immune cell line, just to show it in vitro, but also in glioma-bearing mice. We can see that very specifically the myeloid cells, so the cells that come from the spleen, etc., they have a lot of perfluorocarbon on board, while lymphoid cells, so T cells, etc., don't really. So this is what you also see in other diseases, so it's good to see it confirmed. But interestingly, also non-immune cells take up some perfluorocarbon to some degree. And then we see mostly that in these myeloid cells, that tumor-associated macrophages make up a large part of the immune cells. There are some monocytes, so that came from the outside, so and infiltrated there. They carry some, but there's again no neutrophils that take up the perfluorocarbon. And then we're currently doing some RNA sequencing of these rhodamine-positive tumor-associated macrophages to see what pathways of these immune cells have been activated. Then I've been talking about monitoring, right? So what can we do with that? So in one of the interesting observations that they made in the Joyce lab was that the balance of the type of immune cells before and after irradiation changes a lot. So if you have control animals, then you see that there's a lot of microglia that are present, and then past irradiation, there's a lot of infiltrating cells from the outside, and this goes back down to a lot of microglia being present. So since people are thinking of developing immunotherapies, an interesting thing would be, indeed, can we stimulate more macrophages and etc. to come and attack the tumor over time? So we want to check this, if we could detect a similar pattern with fluorine-19 MRI, and we could. So here you see the perfluorocarbon concentration over time, basically. So here is just before irradiation with of the tumor, and then 7 days after, there is a higher concentration of perfluorocarbon, probably because there's more macrophages present, and this goes back down again over time. Then this very interestingly, and this is very recent, so we haven't fully processed this yet, but here you see one mouse. You see different slices of the tumor, and we wanted to see what happens if we inject two different perfluorocarbons, so one before the irradiation, so that's PFC number 1 here, goes to a small part of the brain, a small part of the tumor, and then if you inject a second perfluorocarbon after the irradiation, so that should label a different biological process than the first perfluorocarbon, and indeed we could see that the second perfluorocarbon really goes to a different site in the tumor after the irradiation. So what we can see here is that with two different perfluorocarbons, we can really detect two different biological processes. And this is possible because different perfluorocarbons have different resonance frequencies. So we can just switch the frequency. They are very far apart, like on the order of 6,000 hertz or so at 3 Tesla. So we can easily just switch that frequency and not detect the other compound at all. Then, just to make a little sidestep before we dive into the clinical part of fluorine MRI. So there's a continuous drive to make fluorine MRI more sensitive. Here, for example, in the multiple sclerosis model, EAE, which some of you probably know, if you do this even at 21 Tesla at the room temperature, you see very little inflammation. Fluorine MRI is just not sensitive enough to detect this, and not enough fluorine MRI can perfluorocarbons can go through the blood-brain barrier. However, if you, for example, use a cryoprobe, then you can boost the signal that you can see that yes, there are particles that actually made it through. It's just not a lot of them. So this slide was given to me by Sonja Weitjes from Berlin. So and this very nicely shows that if you use a high enough field strength and you use a cryoprobe, for example, then fluorine MRI can become quite sensitive. Then another thing that's going on a lot is more really on the chemistry side. So people say, okay, if there is not much fluorine signal, then maybe we can attach more fluorine to each molecule or each particle, and then that way deliver more fluorine. So here is a particle that very shyly is called Perfecta by an Italian group. But as you can see, it contains a lot of fluorine, and they're all chemically equivalent. So this particle creates a lot of signal. Similarly, you can develop polymers, as you can see here, or silica nanoparticles, as I was involved in with a group in Quebec, where you can also, in these holes in these silica particles, you can also put medication. So there's also a large drive from the chemistry side to develop more sensitive particles with a better biological profile, etc., etc. But so how to get fluorine MRI now in the clinic? We've shown that we can detect inflammation. We've confirmed that indeed it's in the immune cells, and that it's safe, etc., etc. When I asked this question to Mangala Srinivas, who was at Radboud in Nijmegen and now is part of the company Senja Imaging, I got sent this this slide basically on the clinical grade manufacturing of nanoparticles. So in the beginning it looked kind of okay, you need to go through a few steps, but then when I clicked this slide, indeed there was a lot of stuff that was appearing. Basically all these steps need to be followed if you make a new particle and want to get it through through the necessary authorities to get full approval basically and to start your clinical trial in the end. So yeah, nobody or very few people without industry support can do this by themselves. So if me as just a lecturer at the University Hospital wanted to do this by myself, I would need to make some shortcuts. So just to put it in perspective, there has been one very small trial of Fluorine Mri. The group of Eric Ahrens already in 2014 labeled some dendritic cells with perfluorocarbon, and oh, my apologies, and then injected these in stage 4, so end stage basically colorectal cancer patients, and saw indeed a signal. But that's the only thing that has been done in humans that with MR basically so far. This was like 3 patients only, and they basically showed with a surface code that they could detect the signal, and that's it. So there's more to be done to make that clinical translation. So what do we need if we group this all together? We of course need a unique application that clinicians actually want, and that need has something advantageous compared to existing regular MRI, PET, SPECT, ultrasound, etc. We want high specificity and high sensitivity. Well, those should be kind of intrinsic to Fluorine MR, but although the sensitivity isn't all that, so we need to work on that. We want a regulatory approved compound, which normally means toxicity trials, quality control, clinical grade manufacturing, etc., etc., and a regulatory approved protocol, meaning personnel training, ethics committee, etc. So how can we, well, either quickly bypass or go through them in an efficient way? So I'll talk a little bit about this in the remaining time. So one of the main troubles of Fluorine MRI is its clearance. So regulatory bodies like the EMA, Swissmedic, or FDA, they don't like it when stuff sticks around to the body for a long time. And that means that you need to have small perfluorocarbons, and small means not a lot of fluorine on them. And if you look at this curve, basically, which is molecular weight versus organ retention half-life in a logarithmic way in this sense, then yeah, we can't use all these massive particles that people have developed because they will just stick around too long. There's especially one that's very low on the retention time, so that regulatory bodies might like a lot, and that's perfluorooctylbromide, which I showed in the beginning, and that has that bromide group in there somewhere, which means that all the resonances are inequivalent. So this one gives a headache on the MR side, but it's very nice for retention. And this is a common decision that you need to make with Fluorine MR and new compounds. Is it good enough to be injected, but is it also MR favorable? Well, even if you have these unfavorable particles like this perfluorooctylbromide, you can basically do something like FET suppression, so Dixon imaging, like to get rid of all these resonances. So here are the resonances of this perfluorooctylbromide. So the CF3 group has a nice singlet, as does the bromide group, but yeah, there's only 2 fluorine and 3 fluorine here. And then there's all these signals of these J-coupled middle fluorine groups that just cause a mess. So when you take a look at a little phantom like the one over here, and you then make a simple image of this with a gradient echo sequence, then you see that all of them ghost a lot. So here I had the resonance on the CF3, so the brightest signal here, you can see nicely follows these little tubes, but there's a lot of ghosting. And then when you use different echo times, etc., these ghosts all move around, etc., and cancel each other. And so if you would ever do this in vivo, you wouldn't see much. So then by doing something like fat water separation or similar imaging, which we did together with Diego Hernando in Wisconsin. You can basically model the signal of each of these resonances because they have a known ratio, right? And you can kind of fold them back onto the same pixel as where they came from originally. For this, you need a multi-echo acquisition, but that's fairly straightforward these days on most scanners. So we developed a technique that we could use for imaging perfluorooctiobromide with high sensitivity. And when we compared them to this one to the ones that are around, so UTE imaging or multi-echo, tailspin echo, then we could see that our technique, especially the SFP version, was more sensitive than the other ones. So that's quite promising for clinical applications, quite straightforward to do this in practice. Then everybody's been doing compressed sensing, of course, the last 10 years. So we also had to jump on top of that. But since we didn't know too much about it ourselves, we asked the group of Tobias Kober at Siemens for some help. And indeed, this is work done by Emeline Dassault. You can see in this mouse, and if you inject a little bit of perfluorocarbon and put a reference tube next to the animal, just to make sure that you see something, then you see almost nothing where the liver and spleen are. And then the big idea was, what if you undersample this image, like what you need to do for compressed sensing, but preserve the K-space center where most of the signal strength is anyway, and then use that gain time, so all this undersampling, to average everything a little bit more, though, and especially average those K-space coordinates that already contain a lot of signal. And then indeed, we saw that this works quite well. So this is a kind of combination of the denoising effect of compressed sensing and the motion robustness of averaging that we have here. So this free breathing mouse, in this free breathing mouse, we could very nicely show the liver and spleen that we couldn't detect at the same time with regular imaging. Also, if we just tried denoising of this standard image through some by switching into the wavelet domain and back, that image didn't look as good as this undersample and average. So quite paradoxical, but it worked, and it will probably help us out in the clinic as well. Then we did a lot of sensitivity optimization, so just finding out for given temperatures, conditions, etc., what the best acquisition parameters are. So this was work done by Roberto Carlotti during his PhD. And indeed, now no matter what we inject, which compound and so on, we can now easily calculate what sequence parameters get us the best signal, the most signal basically for a given unit of time. Then we wanted to show that what we do did at 9.4 Tesla with the atherosclerotic plaque. So these are mouse atherosclerotic plaques that have a volume on the order of 0.1 cubic millimeters or so, that we could also detect them even at the clinical scanner. So this is with a 3T clinical scanner. And yes, we could indeed nicely detect the atherosclerotic plaque in these animals as well on the clinical scanner, showing that sensitivity should not be a threshold or a hurdle for Fluorine MRI on the clinical MR scanner. Then I want to show another slide from the Düsseldorf group, which did some scaling up. Basically, they used what do you call it? A pig model of infarction. So basically in these pigs, they inject a little balloon down the coronary artery and cause an infarction downstream. And then they imaged the heart later ex vivo. So they didn't want to be bothered by surface coils and the beating of the heart and the respiration, so that they just took the ex vivo heart and imaged it several hours in one go. And then they very nicely saw that if you look at a single slice here through the infarct, this here is the late gadolinium enhancement image. So late gadolinium enhancement shows basically retention of gadolinium after a while. And in this late gadolinium enhancement thing, you see basically scar formation in the heart. And then at the same time, they did the fluorine MRI, and you can see that the two overlap, but not completely. There's a region where there is scar formation, but there is no fluorine MR yet. So you can see really different types of processes going on at the same time. And here's the confirmation on the right in histology. So here's the stain for collagen, basically, so scar again. Then here's a stain for immune cells, and then you can nicely see indeed that this region here does not overlap between the two. So really showing also at large scale that you can use fluorine MRI to in principle image inflammation. Then one of the bigger hurdles, indeed, as I mentioned before, is the compound that we're going to use. And this was indeed, or has been, everybody's biggest headache. How are we going to get a compound approved in humans without going through toxicity trials, et cetera, et cetera? And of course, the answer, which I was kind of lucky to find out, is to use already approved compounds. So I found a small startup company that has used this perfluorocarbon called Oxyside, or ABL101, in clinical trials to oxygenate traumatic brain injury. So when people have had serious accident and there's a region of the brain that can't receive blood, you can inject perfluorocarbons because it is a much smaller particle size still than red blood cells. So oxygen that they carry can reach the injured brain. However, they had never heard of fluorine MRI, so there was kind of like a match made in heaven. So I asked them if I could use their compound for MR, and very nicely also these 9 fluorine atoms here on the top, they're all magnetically equivalent, so there's a very nice big singlet that's very far away, as you can see in the chemical shift, from the mess that these J-coupled other fluorine atoms have. So it's also an easy imaging compound. You can kind of use the same pill sequences that you can use for proton imaging, very easy. And furthermore, it has a nice fast clearance from the liver and spleen. So this was more research done by Emile Dassault. So essentially you can see the liver here. And then if you look a few weeks later, basically there is no signal left in the liver and only a tiny bit left in the spleen. So the clearance is quite fast, as you can see here in these curves. So this indeed, also from a clearance point of view, should not be too hard to get to Swissmedic. Actually, it has already been used in the past for these traumatic brain injury trials here in Switzerland, in the Ticino. So it is looking very positive. So with that in hand, I went to the SNF and asked them for some money to maybe do the first fluorine MR trial in humans. And they said yes. So that's the PhD that Simona Rumak started. And basically in this project, we will combine all these tools that I showed before. So we will image, again, inflammation and atherosclerosis, so the carotid artery plaque. That's kind of nice. The surface coil can be put very close to the carotid artery, and of course regularly surgeons take out these atherosclerotic plaques, so we can use histology on these endarterectomies as a ground truth. So we now have a big team together with Patrick Michel from neurology, Sam Rotman from pathology, Thierry Buclen from pharmacology, and François Saucy from vascular surgery. And with this team, indeed, somewhere later in this year, we should start recruiting for this trial where we want to image inflammation and do some parameter mapping in the carotid arteries. As a start and as a last slide, indeed, just to show that something is going on indeed in this already, we bought the Fluorine Mri Coil. As you can see, it's placed here in the neck like this, and an EPFL student, Eliane Brechtbühl, she basically characterized this coil and was able to show with the custom designed phantom where all these different holes have different concentrations of perfluorocarbons. Indeed, the hole diameters are also different. And she was able to show then, yeah, you can image it with proton imaging with this coil, but you can also image it with fluorine, and you can correct the intensity. And then if you plot the concentration versus pixel intensity, you get a very nice fit with a very high R-square value, meaning indeed that we will be able to quantify the perfluorocarbons in the neck very accurately versus a reference with this RF coil. So that's just the very start of this clinical trial, but it should happen soon. So with that in mind, my take-home messages for today are that indeed Fluorine MRI is a background-free modality that allows for the quantitative monitoring of information, cell tracking, and molecular imaging. Its sensitivity is continuously improved through new imaging techniques, new particles, and new hardware. And translation to the clinic is challenging, but should be achievable with the right tools and the right people in place. And several trials have recently started to have academic funding and industry involvement. Thank you very much for your attention, and I'll happily take any questions.
Yeah, thank you very much, Ruud. It was very impressive, very nice talk. I really enjoyed it. Congratulations.
Thank you.
I just had a question that came up in my mind. You certainly thought about it, but I don't know if it's feasible. Concerning the increased sensitivity of PF4B imaging with this multiple resonance, did you think of doing just the opposite, playing with the low bandwidth and to try to separate chemical shift displacement in order to recombine the image?
Yes.
Yes.
Yeah, so there's the two ways that has been done actually. So the simplest way indeed is just select a single resonance, focus on that one, and have it completely uncoupled, so nice high relaxation times, etc. And just focus on that single resonance. But then you're only left with a few fluorine atoms per molecule. That's been done. And the other technique indeed is what is developed, I can't remember where they indeed do a kind of multi-echo or multi-slice turbospin echo. But instead of doing multi-slice, instead of different slices, they do different resonances. So you acquire with a low bandwidth one resonance, and then while you're waiting for your TR to complete, basically, you pick the next resonance and focus on that one, acquire 2D or 3D, some slice and so on. So this is kind of a multi-chemical shift way. However, that works well at high field strength because all resonances are nicely separated. But at 3 Tesla, they're a little bit too close together to do this kind of multi-resonance and then combine them thing. But that works well at high field, so at 9.4 and so on, that's fine. But at 3 Tesla, the resonances of PF4B at least are a little bit too close.
Okay, thank you.
Are there any other questions? I don't see anybody raising their hands, virtual hands as it were. So Ruud, then I'll finish my question and then we'll jump to Mary. So Ruud, I think the fact that you did much of this at 3T makes it ever so much more meaningful because it's very close to the patient, right? I don't think we are ever going to do a patient at 20 Tesla and with some, how did you call it, cryoprobes injected. So I think this is a losing battle for clinical research. However, it might be interesting for basic research. So doing this at 3T is very, very meaningful. Then I also think it's a super impressive journey because you kind of didn't let any stone unturned. You tried every which way and you didn't give up when you saw this matrix of troubles coming at you. You know, that the thing you showed how this process would look like from a regulatory point of view. So you kind of found sneaky workarounds to avoid jumping through those hoops. Now, if you think about what you have now, I mean, it's basically given what contrast agent you will use moving forward, right? So that you have very nicely flushed out. Now, the sensitivity issue will probably be a remaining topic for years to come. And you know, it's hard to predict the future, of course, but looking into the future, where do you think major progress will have to be coming from? Let's assume the contrast agent stays the same. So what we can do is we can tinker with the pulse sequences, we can tinker with the coils, and we can tinker with the reconstruction. So where do you see should we put a major emphasis to increase the signal to noise, to be more sensitive, to see into deeper regions of the body, which I think might be important?
Yeah, the answer is going to be predictable, but that's again going to be all of them. There is no single road to victory here. Fluorine-19 MR by itself is a relatively insensitive technique. So there will need to be work on all areas. So from a reconstruction point of view, we can, in static organs, we can go down the road we have. So we just optimize the pulse sequences. We can do some fancy denoising, basically. We can use the highest clinical field strength we can find. 7 Tesla scanners, indeed, would be optimal, for example, since they're now getting more and more accepted for clinical work. But we need, at the same time, to think about pure pulse sequence type things. So if there is some smart way that we can also acquire a little bit faster, we need probably more sensitive RF coils. So we've now seen, for example, for the brain, there are 128 element brain RF coils. I've seen some cardiac RF coils, I think in Utrecht, where they use 128 elements or so. This kind of thing will probably need to be used as well, just to make us as sensitive as possible. So we can't afford to focus, I think, on a single of these areas, and all of them need to be explored. As far as I know, and yeah, in the end, the signal is just related to the amount of perfluorocarbons that you have present. You can detect them a bit more sensitively with RF coils and higher field strength, etc., and optimize what you detect with pulse sequence. But that is it, I think. So I don't think we'll see some massive leaps or anything in this sensitivity, but there will still be improvements over time. But I think just having this approved compound with a single resonance, that helps us enormously already by itself.
Thank you. Pino, until when is this session? Until 5 minutes ago?
No, you can just keep going.
Okay, so Mary raised her hand. Mary, the floor is yours.
Thank you, Ruud. Super nice presentation. I found it very interesting. It's quite far from what we do, but not so far, because of course I was listening to you on quantitative and inflammation and so on, and I was thinking about multiple sclerosis, and then you put the slides. So super nice that you presented at least one dedicated. And this is my question, because in multiple sclerosis, as you know, so this is autoimmune inflammatory disease of the central nervous system. So inflammatory is everywhere, and we are always trying to keep track of these active lesions, and we do that with MR, with advanced biomarkers at 3T or at 7T. And I was wondering, to your knowledge, if so we do these advanced biomarkers based on microstructure imaging, based on diffusion, or as well based on 3D epi sequences, T2 start phase contrast, basically depicting iron and things like that around the lesions that indicate us for us that this is a lesion that is being active. To your knowledge, on this on your marker and on these mouse models, this has been compared to these other sequences that are depicting inflammation?
Yes, so in the study where they did this mouse model, yeah, they did indeed a few other modalities in there as well. The main problem of fluorine, though, is that you need these nanoparticles, right? And they don't go through the blood-brain barrier very easily. So the brain is probably the worst organ by itself to look at with fluorine MR, because indeed you need a broken blood-brain barrier. We're able to do the glioblastoma imaging because indeed these glioblastomas, they break down the BBB as well, and they become kind of porous around there. But it's not really for more, say, subtle diseases, such as indeed, for example, early stage multiple sclerosis, you probably wouldn't see anything because the particles can't get through, can't get there. Okay. So it is nicely possible to follow multiple sclerosis over time, as I've seen these studies by other people, because in these mouse models, it really starts in the tail, or in the spine, and it works its way up from the spine towards the brain. I'm not sure how this goes in humans, but basically you can nicely track indeed that the fluorine signal starts to appear in the end of the spine, and then it goes up basically through the spine towards the brain in these models. We tried this a little bit ourselves as well, just with our 3 Tesla, with a group in Geneva that has a mouse model of multiple sclerosis, which is, by the way, a very finicky model. It's quite hard to reproduce, etc., etc. But we were not able with our simple protocol at the time to see anything in these mice. Basically, we were also having a tough time handling this mouse model. But a quick exploratory study that we had back in the day, we were not even able to say to see anything. So to be very honest, I don't think that Fluorine MR is the ideal modality to look at inflammation in MS.
Okay. Thank you.
Okay, are there any other questions? So I think, Ruud, with this research, you are, I predict you are on the threshold of a real high impact publication there. If you really can bring this to the human setting, even though we are not supposed to measure impacts anymore of the journals. But nevertheless, I think this could have a major impact, bringing this to the human setting and taking it from there, because inflammation is so important at so many different levels of organs in the body.
Yeah, the main question indeed is now, well, I don't think anymore that the regulatory thing, which I'm writing, by the way, I should be submitted by the end of the month or so, should be a serious hurdle. I think the main problem is going to be recruitment in these patients, right? Because there's a limited number of patients with that get endarterectomies. So that will be the main challenge, to keep in contact with the clinical team all the time and really be on top of this and try to recruit these patients that are willing to undergo this procedure. So that'll be, I don't need that many to show feasibility. But then again, there aren't that many procedures that are being done. So that'll be an interesting hurdle by itself indeed.
So I see John Prior is on the call, on the meeting. Now, why not doing this with PET? Wouldn't it be a heck of a lot easier just to do the same research with PET?
Yes, this is something I was going to propose, and I was after the meeting going to contact Ruud to ask this, because I think it is a large enterprise what he's doing, and having PET looking at sodium fluoride could be something interesting to add. It's going to be so hard for him to put everything in place, and this would be just one more examination. But we know for patients, it's also an additional hurdle to go into another scanner, make another appointment, and so on. So this is something to see, but I would be really interested into looking at our tracer, which is already accepted on the market. And this would be something that could be quite interesting. We can also try to look for financing differently, but I believe this could be probably additional information. I don't think we measure the same exact thing, and that from the scientific point of view would be very, very nice. So again, congratulations to setting up all that research project, and we would be more than happy to participate into adding one more examination, which I hope will not afraid patients.
Yeah, thanks a lot indeed. Yeah, PET of course is the gold standard for molecular imaging, so we'd be very happy to add that as well. Indeed, a good question is indeed if it scares people by itself, but there will only be a big injection and they will have surgery. So as one extra layer, probably it should be manageable. But indeed, it would be good to meet on that subject. Indeed, yeah.
I'd be happy. Thank you for the question, Matthias.
Thank you very much. Are there any other questions? I'm just trying to switch back and forth to see hands and names who I could call on. No more questions. Then thank you very much, Ruud, again for this wonderful presentation, which is really a stellar example of what's going on in the CIBM, not only right now, but also what's happened in the past, and looking forward into the future of the many successes that the CIBM will have, I think. And with that, I would like to hand over to Pina.
Yeah, thank you very much, Ruud.
Yeah, thank you, Ruud, and thank you, Matthias, for chairing this seminar session.